Hello everyone, you are welcome to biology video lectures by Akerele Oladimeji Philip. How has been your week? How is the reading and studying going? On today's edition of the video, we're going to be having fun. It's just going to be the continuation of the series we've been having on cell. So today is going to be cell and its environment. Alright, and this will be our flow charts. These are the factors. Now, to start with, if you've seen a video on cell, we've explained parts of a cell, the cell membrane that is semi-permeable, and some other organelles it has. Now, these factors listed here are basically the processes that explains how the cell reacts to its environment. All right, so I'm very sure most of us are familiar with all of these before now. So let's get started. Now, before we get started, there's something we need to understand. You know, the environment of a cell is most times, if not all of the time, liquid. And that liquid has different concentration, which has been explained in 3 words: hypotonic, hypertonic, isotonic. Alright, so let's now check what does it mean for a cell, what does it mean for a solution to be hypotonic, isotonic, or hypertonic. And let me say at this juncture that a solute is a substance that can dissolve in something, can dissolve in a fluid. Alright, now the fluid that has the capacity to dissolve a substance is called solvent. So the mixture of solvent and solute forms solution. Having established that, so let's now talk about types of solution. You can see here isotonic. Iso means same, tonic, I'll talk about the concentration. So in this case here, what it means is, look at this cell here. This around place is its environment. Now, looking within the cell, the solute are the brown substances, alright, and I don't know what color that is, like green, army green. This brown structure is also the solute outside. Now, according to this diagram, 20% solute is inside, 20% solute is what? Outside. So the same solute concentration inside the cell and outside the cell causes the cell to have an equilibrium. So nothing goes out, nothing comes in. Do you get what I'm saying? Because as it is now, water does not go out, water does not come in. Because this is the point. Once there is imbalance in the environment, meaning imbalance from the outside to the inside, water will go to the part that is more has more concentrated. That's the law. You soon get to say that. So, but because there's balance between here and here, water doesn't go in or out. It goes in, then it goes out again. So it's equal. Now moving to hypertonic. Look at the cell. Look at the inside the cell. The cell is having some few solute, but the surrounding is surrounded by, so is filled up with plenty solute, which means that this cell is having lesser concentration compared to this. So like I told you earlier on, I said wherever you have imbalance between the two environments, the water will go to where it's more conc. Looking at it, look at outside, 40% solute concentration, 20% solute concentration for the inside of this one. So apparently water will come out, so this cell will shrink. Now moving on, in this case here, see the solutes are inside, solute are outside, but in this case the solute outside are more than the solute inside. Now I mean the solute inside are more than the solute that are outside. So just 2 solute outside, see 20% solute inside, 10% solute outside. So like I told you, water will go to where there is more what? Solute. So inside we have more solute. So the cell will swell, become stodgy. So you can just pause the video and read what we have here. Do you get? Now you can see here it says that water particles will move into the cells, causing the cell to expand and eventually burst. That means lyse. This is only applicable if this cell is animal cell. If it's a plant cell, it will not burst because it has rigid cellulose cell wall. So that's just the background. So it means that an isotonic solution is a solution that has equal, let's say, solute and solvent, so to say. And most of you know that a solution is isotonic or hypo or hyper until it is compared with another solution. All right, so let's get started. So what's diffusion? That's the movement of particles, and that particle talks about fluid, ions, solutes, and the likes, from a region of high concentration to what? A region of low concentration. All right, so look at this. This is the solute here. This is the substance here. Once it was introduced here, gradually it is spreading. See it spreading, until it spreads around totally. That is diffusion. Let's look at another picture. Another slide to explain that. Look at this. This substance was introduced gently into this fluid. Over some time, it has spread around. It's fully dispersed. All right. So this explains diffusion to some extent. So then let's move on to what are the factors that affect diffusion. We have temperature. We have the temperature, the faster the diffusion. How? Because by kinetic theory of matter, substances tend to move faster at higher temperature because their speed is increased. Also, large surface area, surfaces that are very large will definitely create more space for diffusion. Then concentration gradient. Diffusion is always high from where we have higher concentration to lower concentration. That means if you have two environments, for example, you have I'm trying to see how to explain that. You have part A and part B, and you introduce something to part A, and there's something in part B. Once you introduce something to part A, things will easily move. Those molecules will easily move to part B because it won't be colliding with any other molecule there. But let's imagine you have that same thing introduced to part A, it was not introduced to part B, even though in lesser amounts. The diffusion will still take place, but it will be slower because the molecule of that substance in part B will be colliding with the one coming from part A. Alright? Now, size of molecule. The smaller the size of molecule, the faster the diffusion. Yes, that is true. Except for one element called sodium. Sodium is smaller than potassium and is expected to diffuse faster than potassium. However, sodium has hydrophobic property. It gathers water around it so much that it doesn't pass through membrane easily. Once it gathers water around it, it becomes bigger than potassium. This is the only exception to the smaller the particle, the faster the diffusion. Then looking at this diffusion medium, or we can say substance that is involved. If the substance is solid, the rest assured is going to be very slow. So it is fastest in gas, slowest in solid. All right. Now let's move on to, so let's see what's the application of diffusion. Diffusion is what takes place at the alveolus of the lungs. How oxygen goes from the lungs into the blood vessel, and how carbon dioxide goes from the blood vessel into the lungs. If you've seen my video on circulatory system, cardiovascular system, you will have understood this. Is very straightforward. So this is diffusion taking place. All right. So let's move on to active transport. Now, for you to understand active transport, it's better if we can compare it with passive transport. Passive means no energy. Active means with energy. All right. Now, passive is just almost like the same thing we call diffusion, from a region of higher concentration to a region of what? Lower concentration, going through pores, through channels and the likes. All right. Now, Normally when we say down the hill, you can see downhill. If one is flowing, if someone is swimming in the direction of the flow of river, it doesn't even need to struggle. The river just carry the person along that flow. But active transport is a place whereby you are moving substances from area of lower concentration to area of higher concentration. That means you need energy to do that, which is that energy is form of what? ATP. Alright, so you can see you are going up the hill. So you can see that the molecules here are more than here, but we are still moving it towards where it's more, because this one are meant to repel it. That's why we need energy to forcefully take it there. That is active transport. Then this is osmosis. Osmosis is defined as the movement of solvent from area of lower concentration to area of higher concentration through a semi-permeable membrane. Before we move on, if you have written that down, you will take note of solvent and membrane, semi-permeable membrane. So hence, there are three things that are needed for osmosis to take place. And what are those? An hypotonic solution, a semi-permeable membrane, and an hypertonic solution. I take that again. Hypotonic solution, semi-permeable membrane, and hypertonic solution. Alright? So as it is now, from what we have here, look at this diagram. I don't know if it's clear enough. You will see that there's a fluid here, bluish in color. It's another fluid here covering it almost the same level. But the thing is that don't forget that this whole potato is inserted into this water. Alright, so you can see this is water here, this is sugar solution within the potato. See it here. At this instance here, see the level after some hours, quite a number of hours. See the level of the solution within the potato, and see where the water was here. See where it has reduced to. So what we define osmosis to be is from a movement of solvent. What solvent? Water in this case. From region of what? Lower concentration. This area inside the beaker is lower concentration. Inside the potato is higher concentration. So from region of lower concentration, region of higher concentration, through a semi-permeable membrane, which is the potato. So this water here passes through the semi-permeable membrane. When we say semi-permeable, it means it will only allow solvent to pass, not solute. So that's why it is only water that can pass to go and meet the solute, to solution. Then the solution then increases in volume. Alright? So let me show you another diagram to explain that. Beautiful. You see that this is water molecules here. This is sucrose, which is a disaccharide. So this place is a solution. Apparently speaking, this side is dilute. This side is what? Conc. Another word for dilute is hypotonic or lower concentration. Another word for concentrated is hypertonic or higher concentration. So we said osmosis is movement of solution, of solvent, water molecule, from area of lower concentration to area of what? Higher concentration through what? A semi-permeable membrane. Alright? Now moving on, we have some other things there in that flow chart. Let me show you that in a minute. That we are meant to see. We have talked about 3 of those already. Now look at this. We've talked about diffusion, active transport, osmosis. So we'll talk about the 4 of these together with some few diagrams. It promises to be nice. Beautiful. Now I want you to bear in mind that the response of plant cell to solutions is not the response of animal cells to solutions. Alright? Now, in the case of plant cell, they have a rigid cell wall added to their cell membrane, while animal cells don't have at all. This red blood cell is from an animal. Alright? So when you put a plant cell and animal cell in hypertonic solution, see what happens. In animal cell, the entire cell shrinks because water is sucked out. All right. Yes, water is sucked out through the plasma membrane or cell membrane. So the cell shrinks. The process is called crenation. All right. Now, but for plant cell, it becomes plasmolized because it's not the entire cell that shrink. Why? Because the cellulose cell wall is rigid. It doesn't respond to that. So water is only sucked out from the inside. So the entire contents, the plasma or the cytoplasm, the whole thing does what? Shrinks. So you can see the difference. This one, the entire cell shrinks. This one, just the internal environment, the plasma, cytoplasm and the nucleus shrinks. Why does it not shrink? Because the cell wall is made up of cellulose, which is rigid. Putting the same cell in isotonic solution. In isotonic solution, it means that water will not be sucked in or go out because the cell is having the same concentration with its environment. So water will not go out. So the cell remains the same. All right, it still remains the same. For plant cell, it becomes a bit flaccid. That means it reduces a little in size, just a little in size. But when these same cells are put in hypotonic solution, that means a dilute solution, see what happens. For plants, animal cell, it keeps sucking in water until it bursts. Can you see this one is ruptured? Why does it burst? Because the cell membrane is flexible, is fragile, so it will just burst. In this case, because it's red blood cell, we call that process hemolysis. It's one of the processes I've written up there, hemolysis. So, but for plant cell, it won't burst. It will just become so expand and bulging. The process called what? Turgidity. All right. So plant cell is meant to actually have busted if the cell wall were not there. So because the cell wall is there and it's rigid, it doesn't give in to the swelling. All right, it just stretches alone. This is another diagram to explain that. You can just pause this and see, just read everything I've said verbally are all here. All right, so it's very simple. So this will be the concluding aspect of cell and its environment. I hope you've been able to learn one thing or the other. Please feel free to contact me in case you have questions or you want to re-explain some things, or probably you have some advice to give me to make helping you better. Thank you.